Well, thank you to everyone so much for joining us today. Welcome to the first NECAN industry webinar of this series. The NECAN Industry Working Group has gone through a big revival in the last year, and it's currently composed of aquaculturists, extension agents, resource managers, and aquaculture researchers. The group has been working to put together this series of webinars focused on industry-related aspects of ocean acidification and other coastal changes. And our aim is to involve more industry members in these kinds of conversations and also hopefully learn directly from some industry members what impacts they are seeing on their operations. From the NECAN website, you can see the full list of the webinar schedule, which goes through December. And today we'll be hearing about the clamming industry and things that have affected them so far. So the clamming industry has been experiencing declines in landings. And today we'll hear from two researchers who have been investigating the potential role of sediment saturation state on clams. Dr. Jeff Clements received his PhD from the University of New Brunswick, St. John, where his research focused on the influence of sediment acidification on burrowing behavior, post-settlement dispersal, and recruitment of juvenile softshell clams. He went on to work as a postdoctoral fellow at the Department of Fisheries and Oceans, Canada, and he will soon start a Marie Sklodowska-Curie postdoctoral fellow position in the Department of Biology at the Norwegian University of Science and Technology. And then our second speaker is Dr. Brian Beal, who is a professor of marine ecology at the University of Maine at Machias, and is also the director of research at the Downey Institute for Applied Marine Research and Education. He has been involved in fisheries and aquaculture research in Maine for several decades, including applied research projects with lobsters, sea scallops, quahogs, surf clams, softshell clams, and green crabs. So thanks so much to both of our presenters today. Each presenter will have 15 to 20 minutes to give their presentation, and we'll open it up to questions and answers at the end of both presentations. You can submit questions through the GoToWebinar control panel. Emily can correct me if that is incorrect. Or at the end of the period, there should be a function to speak your questions during the question and answer period. So, excellent. We will get started with Jeff's presentations on sediment acidification effects on bivalves and their ecological consequences.
All right, thanks very much, Meredith. So I see there's about 40 people, a little more than 40 people on board. So thanks everybody for tuning in, and I hope I can give you a little bit of information from a lot of my PhD work that I conducted at UNB regarding the behavioral effects of sediment acidification on juvenile bivalves and what the ecological ramifications of that are. So we'll talk a little bit about some things beyond dissolution today. So most of you have probably seen a slide like this. So first we'll just introduce ocean acidification for the folks who may not be aware. So essentially ocean acidification is this process that mainly occurs in the surface of the open ocean as we burn more and more fossil fuels and pump excess CO2 into our atmosphere. So as most of you probably know, the ocean absorbs about 30% of all of the excess atmospheric CO2 that we put into the air. And this dissolved CO2 reacts with water to create a weak acid known as carbonic acid, which inevitably dissociates into the constituents that you see on the right-hand side of that flow chart. And what this inevitably ends up causing is a sort of triplicate effect on animals that live within the ocean. So we get an increased amount of CO2 in the ocean that we call hypercapnia. We get an increase in the amount of hydrogen ions in the seawater, or a decrease in the pH of the seawater, which we typically refer to as acidification. And at the end of the day, we get a decrease in these carbonate ions, which we often refer to as carbonate undersaturation. And these three processes that are driven by the dissolution of CO2 into the ocean can act independently or synergistically to affect animals within this habitat. So sediment acidification, the process is essentially the same, except the cause of sediment acidification, or the source of the CO2 that drives sediment acidification, it comes from a different source. So really, we're going to focus here on the surface sediment. So just focus your attention to where that arrow is pointing. In the surface sediment, most of the CO2 and the acidification that happens comes from aerobic respiration. That's driven primarily by the degradation of organic matter by microbes. So what happens is organic material accumulates on the top of the sediment. Once it's dead and it starts to decay, microbes consume it and they breathe like we breathe. So they use a lot of oxygen and they create a lot of CO2. So this excess CO2 goes into the pore water of the sediment at the surface, and it drives those changes that I just showed you that occur through ocean acidification. So this process has been linked to a number of biological effects, and most of the early work that we look at when we talk about clams is what's referred to as death by dissolution, and it's been pioneered by Mark Green down in Casco Bay for the most part. And what they showed in one of their pioneering studies back in 2009 was that the saturation state of the sediment was far lower than that of the overlying water, and it typically was lower than 1, which is a threshold that we consider pretty important for calcifying bivalves, because an aragonite saturation state below 1 promotes shell dissolution. And that's what the authors of this paper found. So what they saw was that increasing exposure to these acidic conditions in the sediment surface resulted in the dissolution of shells of settling or very young small bivalves, and that drove higher mortality in these bivalves. So in that graph that just popped up, what you can see is the rate of mortality on the vertical axis and the size class of bivalves on the horizontal axis. And then the different colored bars correspond to the different saturation states, with the darker bars corresponding to better conditions or higher saturation state conditions. And what you can see is that particularly for the small bivalves, the lower the saturation state, the higher the mortality. And to further this point, Green and his colleagues showed that if you go out to the sediment and buffer this with crushed shell, at the end of the day, you end up with a higher number or a higher recruitment of settling soft shell clams. So this is one of the earlier papers on sediment acidification effects on bivalves. And since then, we've seen a number of other effects, primarily sublethal effects. So there's been a couple of different effects observed. One, increased heavy metal accumulation under higher levels of sediment CO2 or lower sediment pH, and also increased prevalence of tissue lesions, which can interfere with biological functioning of these animals. Now, another sublethal effect that has become commonly observed in these bivalves is their burrowing behavior. So we've observed this in a number of different species at a number of different life stages, including larval hard clams, juvenile softshell clams, and adult manila clams. And the graph on the right, what you're looking at is the general response of bivalves to sediment saturation state and sediment pH. So again, on the vertical axis, you've got the proportion of animals that burrowed of a given number that started. And on the horizontal axis, the top graph shows pH and the bottom graph shows saturation state. And despite these different species and different life history stages, we see a surprisingly consistent effect. And that effect essentially says that when the sediment is more acidic, less bivalves burrow into that. So in that paper, we interpreted this as a functionally beneficial response. And the reason that we said it was functionally beneficial is that if bivalves don't burrow into low pH or low saturation state sediment, then they may be able to avoid death by dissolution by relocating themselves elsewhere in a more optimal habitat. So what I want to do for the rest of this presentation is really dive in a little bit into these burrowing behavior responses and outline some of the nuances that we've observed, or that I observed during my PhD, within this sort of realm of sediment acidification. So with respect to juvenile softshell clams, where does the evidence come from that they don't burrow into low pH or low saturation state sediments? Well, our first study really happened in the lab. And essentially what we did is took some sediment from the field, brought it into the lab, and just essentially bubbled it with CO2, much like Mark Green and his colleagues did. And then we put a defined number of clams on top of the sediment and measured how many of them burrowed and how many of them didn't burrow. So unlike Mark's earlier work, we used bigger clams than he did. So we were using juveniles rather than recent settlers. And we saw this sort of positive relationship between saturation state and burrowing. And we looked at two different size classes, so small juveniles and big juveniles, and we found that size didn't really have an effect. So at least in the lab, when we manipulated the sediment, high saturation state, which would promote shell formation, we saw lots of clams burrowing into those, and the lower saturation state sediments, we saw fewer clams burrowing into those. So that's all and well to show that in the lab, but can this happen in the field as well? And we essentially showed the same thing with the field, only the relationship was a lot stronger. So here, this is from a different paper that we published in 2016. And in this, rather than manipulating the sediment saturation state or the sediment pH in the lab, we just went out and collected a number of cores from the field where we had measured different sediment pHs out there. So we were measuring pH in real time, and we were collecting cores that spanned a gradient of sediment pH. And essentially what we did was place the core in the field and put some clams on those and observed how many burrowed in real time. And essentially we saw the exact same thing that we saw in the lab. So higher pH, higher saturation state, we saw many more clams burrowing into those sediments than we saw in the lower pH and lower saturation states. So again, we said we interpreted this as a functionally positive or a functionally adaptive response, as an avoidance to death by dissolution. So we did a short lab experiment just to look at how many of these juvenile clams were dying under different sediment pH conditions. So on the y-axis or on the vertical axis there, you can see the percent mortality. On the horizontal axis, we've got 3 different pH treatments. When you think about the context of ocean acidification, these three pH treatments are much lower than what we would expect by ocean acidification. But these are typical values that we actually see in the sediment. So you can see that these sediments are much lower already in their pH and saturation states than is predicted by ocean acidification. So 7.3 was our control, and that was the mean pH that we observed in the field. And then we dropped that down to 7 and 6.8. And what we were seeing was increased mortality at the lower pH conditions or the lower saturation state conditions. So how do the clams know how to do this? How do they actively avoid burrowing into these habitats? So my very first PhD chapter looking at this, really I was looking at how does ocean acidification influence the behavior of animals? And one of the mechanisms proposed by some guys down in Australia and over in Europe was through this neurological interference. So this work was done with fish. And what they were observing with fish is that when you put them in high CO2 water, a lot of their dichotomous behavioral responses completely flip. So for example, if you take a fish prey species and you put it in an arena with a predator, when it's reared under ambient or normal conditions, the prey will actively avoid that predator, right? That's an evolved response so that it doesn't get eaten and die. But when you rear those prey fish under acidified conditions in the seawater and then expose them to those predators, they do the exact opposite. They swim to the predator and they get eaten immediately. So we see these sort of switches in adaptive behaviors. And as I was reading that, I thought, okay, maybe this is what's happening with the clams, because burrowing is a dichotomous response. You're either going to burrow or you're not. So essentially, what we did was we drugged some clams. So we drugged them with this thing called gabazine. And so the way the mechanism works, first, with the GABA interference under normal conditions, you've got these chloride ions at this ion gradient that typically flow into the cell and keep the cell at resting membrane potential so that the animal does what it normally does. Now, under acidified conditions, what happens when the pH decreases or the hydrogen ions increase is that the animals suffer from acidosis. So to counteract acidosis, what they eventually do is accumulate bicarbonate ions so that they can maintain cellular equilibrium. But as they accumulate that, what happens is that chloride ion gradient at this GABA A receptor reverses. And what that does is switches, it activates, it depolarizes the membrane and causes a switch, or can cause a switch in the behavior. So what we did is drug these clams with this thing called gabazine. And what gabazine does is comes in, and where you see GABA in that diagram, the gabazine binds to that and closes that ion channel so that the flow of ions can't change anymore. So what you would predict then is if the clams drugged with gabazine and have that ion channel closed, if you put them in high or low CO2 conditions, they'll behave the same. If they're not drugged with gabazine and that ion channel is open and acidification affects their behavior, you'll see a difference in behavior. And that's what we saw. So if you look at the graph on the left there, the clams that were not drugged with gabazine, the blue points there, in the low pH conditions, we saw a decrease in their burrowing behavior. But when we drugged them with gabazine, in the low pH conditions, they still burrowed at essentially the same frequency as the clams in the control pH. So this is just some evidence that these burrowing responses aren't necessarily driven by saturation state, but can be driven by the pH of the pore water that affects the acid-base balance of these animals and causes a behavioral switch. So what are some of the ecological ramifications of these behaviors? One of the ones that we tested was predator avoidance behavior, and you're going to hear a little bit about predators in a little bit. So what we wanted to see here is that, okay, in the lab and in the field, we put these clams on the sediment, but there were no predators around. What happens if there's a predator? Are the burrowing responses to sediment acidification still the same? So what we did was some laboratory trials where we exposed the clams to predator cues and some clams to no predator cues, and again measured their burrowing behavior in response to different pH sediments. So you've got the percent burrowed again on the vertical axis. On the horizontal axis, you've got a treatment with crabs, and these were an invasive crabs. These are the green crabs, and a treatment without the crabs. And essentially what we saw was that despite crabs being present, the burrowing responses to sediment acidification were the same. So we interpreted this as a negative ecological consequence for the clams, because if they don't burrow in the presence of predators, their chances of being eaten are much higher. The second ecological function that we took a peek at was localized population dynamics. So again, we said these clams don't burrow into sediments that have low pH or low saturation state, but does that mean that they actively move elsewhere and try to find new habitat? And again, we did this in both the lab and the field. So the graph on the left comes from the lab, and essentially we looked at, under different water flow conditions, how many clams moved away from the sediment habitat of a certain pH or a certain aragonite saturation state. And again, what we saw was that at higher saturation states or higher pHs, fewer clams moved away from that and burrowed into the sediment, whereas at low saturation states, more clams actively moved away. And again, we tested this in the field and saw about the same thing. So what we can see here is that not only are the clams not burrowing, but they are moving away from these habitats, and we assume, or we hypothesize, that these clams are looking for more optimal habitat to settle in. But it's important to remember that there's many interactions that can happen with sediment acidification and these ecological consequences. And one of these is temperature dependency. So burrowing behavior is not strictly caused by sediment acidification. Rather, sediment acidification is one of the many things that can influence burrowing behavior, but temperature can also do that. So in one of my PhD chapters, we looked at how sediment acidification influenced burrowing in the context of temperature. And what we found that is that as temperature increases, the effect of low pH or low saturation state on burrowing frequency decreases. So temperature essentially mitigates the effect of acidification on burrowing behavior. Again, this could be bad or good. So there's more burrowing, which means less predation probably under high temperatures, but also these clams are going to be burrowing into really low saturation state sediments. So the probability of death by dissolution there is going to be higher. So there are a number of different nuances that we have to take into consideration with this stuff. So all of this stuff done experimentally in the lab and in the field doesn't really mean a whole lot unless you can actually show this relationship under natural conditions and determine whether or not it might have an effect. So that was the last chapter of my PhD, and what we did was look at 4 different sites up here in the Bay of Fundy that all really differed in their land use associations. So there were different levels of coastal land use going on at each of these sites. And we went out over the course of 1 year in 2012 and looked at the pH of the sediment and how it actually responded throughout the year. So in that graph in the bottom left-hand corner, you can see that pH generally decreased throughout the year. And the other characteristic of these sites was that they had low predator abundances. And that's going to be something that is going to be important to remember when you're listening to Brian's talk in a little bit. So essentially what we did was went out and measured the sediment pH at these different sites, in these different time periods, and took a corresponding sample of clam abundance. And we were looking to correlate these 2 metrics. So when we first look at this, what we see is a pretty strong relationship between sediment pH and clam abundance at these sites. So you can see again, just as the burrowing response that we observed, when the sediment pH is higher, the sediment saturation state is higher, we typically see a higher abundance of clams. Now it's important to consider when you're looking at this graph that this higher abundance of clams at higher pH conditions may simply be driven by a higher abundance of clams during earlier parts of the settlement season, right? So these lower abundances at low pH may simply be driven by natural mortality that happens throughout the year. So to counteract that, we looked at these first 2 weeks when we wouldn't think that natural mortality would drive clam abundances as much because clam settlement is still occurring throughout this time. And when we just looked at those 2 weeks, we saw essentially the same response. So during those 2 weeks, again, sediment pH was correlated with higher clam abundances. So higher pH, higher clam abundances. And to further demonstrate that point, we just looked at this on a site-by-site basis. And what we saw was that the sites with higher pH typically had a higher clam abundance. Now there's some caveats to point at here. And the main one that we found was that sediment pH was also correlated to sediment grain size. And grain size is known to influence the recruitment and settlement of bivalves as well. So there may be some intricate processes happening here where higher grain size leads to higher pHs, and that leads to higher clam abundance. Or this may simply be a reflection of clams responding mechanically to the grain sizes. We're still not sure about that. So just as a quick recap, we see lethal and sublethal effects. And in terms of behavioral effects, we think that this is an adaptive behavior to avoid death by dissolution, but it does increase their vulnerability to predation, and it is affected by temperature. In terms of knowledge gaps, there's still a bunch that we have to figure out. So our experiment with predators used non-native or invasive green crabs, and we hypothesized that maybe the clams simply just aren't adapted to the presence of these predators yet. So experiments using native and non-native predators may yield different effects. Other abiotic factors aside from temperature that are known to influence burrowing as well: salinity, oxygen, and a slew of other ones as well. We still don't know how these things interact to affect burrowing behavior in the context of sediment acidification. And we need more field observations. One of the things we don't know is how sediment acidification has changed over long periods of time. So this webinar is driven by clam declines over spanning years and decades, but we don't know how sediment acidification has changed over that time. We know that it's really, really variable, even within a year. So it could just be simply that, you know, acidification, sediment acidification is something that these clams have dealt with for a long, long time. And there are other things happening that are causing declines in their populations on the time span of decades. And essentially, when we look at field observations, our study used sites with low predator abundances, and we need to really see how high predator abundances influence this relationship that we've observed. And I think that's probably what you're about to listen to now. So lots of people to thank, and I think we can move on to Brian's presentation.